Okay, so good evening. So I think I will just start the class for today and let others join afterwards. Okay, so I think my slides are visible and I am audible too. So good evening everyone. So my name is Isaac Cheria and today I am going to do the week 5 section of the NPTEL course Cell Biology, Cellular Organization, Division and Process. So we will be discussing about 30 questions for today, like for 10 questions from the current year, uh, 10 MCQ questions from the current year, 10 MCQ questions from the previous year, and 10 questions that I have made and will be discussing today. So I will be asking the questions. You can give a response, and if you have any doubt in between, you can just unmute yourself. You can talk, or otherwise you can just type in the chat box. No issue in that. Okay, so I think we can just move on to the first question then. Okay, so coming on to the first question. The significant outcome of meiosis is increase in genetic variation, reduction of chromosome number by half, formation of gametes, all of the above. Which might be the answer? The significant outcome of meiosis. Okay, so I think I will just give you the answer. So answer to this question is all of the above. So basically, if you look into option number one, it is right, because if you're looking into a process called crossing over that is taking place in prophase 1, the packeting stage of prophase 1, what happens is that the exchange of chromatid arms take place between two sister chromosomes, right, from the father and one from the mother. So in that case, there is a genetic variation that is happening there because of the transfer of chromatid arms from one chromosome to the other. So that leads to increase in genetic variation. Reduction of chromosome number by half, yes, because the ploidy at the end of meiosis 2, if the ploidy at the start of meiosis 1 is diploid, then the ploidy at the end of meiosis 2 is always haploid. So because the chromosome number is reducing to half, so in that case there is a reduction of chromosome number there. And formation of gametes, yeah, basically these are involved in the formation of gametes, right? Not in case of like what happens in mitosis. Meiosis happens in case of germ cell, which leads to a formation of gametes there. So the answer to this question is option number 1, that is increase in genetic variation. Option number 2, reduction of chromosome number by half, and option number 3, formation of gametes. That is all of the above. So anyone has any doubt regarding this? Okay, so I think we are clear with this question, right? So I will just move on to the next question then. Coming on to the next question. The order of subdivisions in prophase 1 stage is as of first one of leptotin, zygotin, packeting diakinesis, leptotin, packeting, zygotin, diplotin and diakinesis, leptotin, zygotin, packeting, diplotin and diakinesis, leptotin, diplotin, zygotin, packeting and diakinesis, which may be the option. Which is the correct order of all the subdivision or subphases of or substages in prophase 1? of meiosis 1 stage. So this is the stage where crossing over synaptonemal complex formation take place, right? So there should be substage that is happening there and should have particular order there. And it follows this order, then afterwards once it completes it enters into metaphase 1 and it proceeds into afterwards telophase 1 and then into meiosis 2. In case of prophase 1, which is the order, correct order through which it is followed. Okay, anyone? Okay, so I will just give you the answer. So the answer to this question is option number 3, leptotin, zygotin, paccatin, diplotin and diakinesis. So this is the basic stage, right? So this is the prophase 1 stage. You can see that it starts with the leptotin stage. In that case, the nuclear membrane is intact there. You can see that the replicated chromosome starts to condense in that case, and then it enters into a zygotin stage. So in this case, what happens is that a structure called as bivalent starts to form. So in that case, you can see that this is one sister chromatid, this is the other sister chromatid, right? So both have the same chromosome number in that case. So what happens is that there is a formation of a from the chromatid end. You can see that there is a formation of what you called as a synaptonemal complex. So this extends out throughout the chromosome, and this process is actually called a synapsis, and it begins within the zygotin stage. So in case of paccatin stage, the synapsis formation is complete, and the synaptonemal complex is formed throughout the chromosomal axis. And what you call, and here this particular structure is called as a bivalent because the two sister chromatids are joined to each other, and this particular structure with the help of a synaptonemal complex, and this particular structure is called as what is called as a bivalent. So that happens in paccatin stage. And what happens is that in also case of paccatin stage, the chromosome, as the chromosomes are close to each other, chances are that the chromatic ends of the chromosome actually starts to cross over with each other. So in this case, if you have seen the red chromatid arm, right? So afterwards, when crossing over happens, this moves into this chromatid ends, and the blue chromosome arm at this end moves on to the red one. So this particular structure is also called as a bivalent, and during this process, crossing over takes place. So you can see that in the next stage, that is the diplotin stage, the chromosomes are separated to each other, and some portion of the red chromosome moves into the blue chromosome, and in case of the blue chromosome, it moves into a red chromosome. And afterwards, once it has completed in the diplotin stage, the synaptonemal complex starts to dissolve out. And afterwards, the prophase 1 is ended, and the nuclear membrane starts to fragment so that it can enter into the metaphase 1 of meiosis 1 stage, and then proceed afterwards into the subsequent stages. So these are the particular stages, that is, it starts with the leptotin, where the chromosomes condense. In zygotin, the synaptonemal complex starts to form. In the paccatin, the synaptonemal complex is formed, and particular structure which is called as bivalent is seen observed here. And afterwards, which is followed by process called crossing over. Then it moves on to a diplotin stage, where the crossing over has ended, and the synaptonemal complex starts to dissolve out. And afterwards, it moves on to a diakinesis, which is where the nuclear membrane starts to fragment out, which is so that it can enter into the metaphase 1. So the order is leptotin, zygotin, paccatin, diplotin and diakinesis. So anyone has any doubt in this, please? Okay, so I think it's clear. So I will just move on to the next question then. So coming on to the next question. What is the role of crossing over during meiosis? It reduces the chromosome number. It increases the genetic diversity. It ensures the formation of spindle fibers. It duplicates the DNA, which might be the answer. Okay, I'm getting option number 2. Any other option? Okay, I'm also getting option number 3. Role of crossing over during meiosis. Okay, so I will just explain the answer. So basically the answer to this question is it increases the genetic diversity crossing over. So basically if you look into a previous diagram, you can see that crossing over actually happens within the bucketing stage, right? So in this case what happens is that you can see the chromosome end from this particular sister chromate is getting transferred into this particular end, and the chromosome end from this particular end that has been transferred earlier is moved on to this particular end where it was lost there, right, by a process called as crossing over. So sorry, the process known as crossing over. So what happens is that when a particular gene sequence from one sister chromate is transferred to other, chances that the DNA there is certain genome sequence difference that is happening within this particular end of this chromosome, leading to genetic diversity there. It is similar to what mutations are, right? Because when you are receiving this particular chromatid ends will more or less similar to each other, but they are from two different individuals, right? From father and one from mother. So if consider this as father chromosome and this as mother chromosome. So in this case, if you are receiving a particular sequence from the father chromosome, as you can see here, then some of the sequence will be different there. Similarly, in case of the, you can see that in case of the mother chromosome, if you can see that, you can see some of the sequence are getting from the father chromosome, right? So in that case, the sequence difference is getting exchanged there, and that's lead to genetic diversity there. So it is similar. That's why I said that it is similar to what you see in case of a mutation where genomic sequence can lead to diversity. In this case, the crossing over is the process that is taking place between two sister chromatids or homologous chromosomes, and because of their homology with each other, sequence similarity at certain points of the chromosome with each other, that's why crossing over is taking place. You might have heard about whole junction formation and all that leads to recombination. So homologous recombination take place, and for which results in the this phenomenon called as crossing over or crossover, which in turn leads to increased genetic diversity. It reduces chromosome number, that is right, but it is an overall process of meiosis, right, not of crossing over. The individual process of crossing over is to generate genetic diversity, but it doesn't lead to what is called as reduce the chromosome number. So in the chromosome number is reduced in case of the at the end of the meiosis 2, where the chromosomes are divided into, chromosome number is reduced to half. That is, if it starts with the diploid cells at the meiosis 1, then it ends with the haploid cells at the meiosis 2, right? So it is not a function of crossing over, it is actually a function of meiosis 2 at the end of the meiosis 2. And it ensures formation of spindle fibers. No, it doesn't ensure formation of spindle fibers. That's all done by specialized structures called kinetochores or centrosomes, etc. Right? So centrosomes, right? It duplicates the DNA. No, that happens in S phase. That's already happening earlier also in this case. So that's not the answer. So the answer to this question is crossing over increases genetic diversity. So anyone has any doubt regarding this? Okay, so I think we are clear on this, right? So I will just move on to the next question then. Okay, coming on to the next question. Which of the following statement is correct? Meiosis 1 starts with one cell, then if it starts with one cell, then at the end of the meiosis 2 it ends with two cell. If it starts with meiosis 1 starts with two cell, then it will end up with 8 cell. If meiosis 1 starts with two cell, then meiosis 2 ends with 4 cell, or none of the above. Which of the statement might be correct out of these three statements or four statements? Read the question carefully and then you can answer. Okay, I am getting option number 1. Any other option? Read the question carefully, you have time. Okay, option 3 I am also getting. Option 4. Okay, so I will just give you the answer. The answer to this question, sorry, sorry for that. Answer to this question is option number 2. I will just correct this out. Meiosis 1 starts with 2 cell, then meiosis 2 ends with 8 cell. So in this case, if you look into option, like if I say that it starts with 1 cell, so it's the basic thing, right? So if it starts with 1 cell, you can see that in the meiosis 1 there is 1 cell there. At the end also you can see that 2 cells are formed. So in case of meiosis 1, if it starts with 1 cell, then it leads to a formation of 2 cells there, right? Then it enters into meiosis 2. So in that case, these 2 cells actually, each individual cell actually divides into 2. So you can see that from this one cell it divides into 2, and from this other daughter cell it divides into 2 other daughter cell, giving rise to a total number of 4. So if you start from the individual one cell there, right, from one cell, in case of meiosis 2, at the end it leads to a formation of 4 daughter cells there. From one cell that is entering into meiosis 1, at the end of meiosis 2, 4 daughter cells are present there. So simple multiplication, if it starts with so in this case you can see that if you look into first option, meiosis 1 starts with 1 cell, then meiosis 2 ends 2 cell. No, it ends with 4 cells there. One cell at the end of meiosis 1 divides into 2, and those enter into meiosis 2, leading to a formation of 4 daughter cells there, total of. So if it was 4 cell, then I can agree with that. In case of option number 3, if you look into meiosis 1 starts with 2 cells, if it starts with 2 cell, then it should end up with not 4 cells, right? In case of, if you start with 1 cell, it ends with 4 cell. If it starts with 2 cell, then it ends up with 8 cell, right? So that option is wrong also. So option 2 is the right answer. So if you start with 2 cell, then by simple multiplication you can see that it will end up with 8 cells there. At the end of meiosis 2, it will end up with 8 cell, not with 2 cell or 4 cell in this case. So this is the answer, meiosis 1. If there are 2 cells that are starting up meiosis 1, at the end of meiosis 1 it forms about 4 cells there, and at the end of meiosis 2, if each and every cell enter into meiosis 2, then it forms 8 cells. So option number 2 is the answer. So some of the question will be like in different cases, so I thought that this question would be helpful. So is this clear, or should I explain it once more? If it is 1 cell, 4 cells are formed. If it is 2 cell, then 8 cells are formed. If it is 3, then you know the answer, right? The 3, like at that time you can say it will be 12, right? 4 into 3, 12. Anyone has any doubt? so I think I will just move on to the next question then. Coming on to the next question. Which of the following events occur only in meiosis and not in mitosis? Okay, so anyone has any doubt? Someone raised a hand. Okay, okay, good. So coming on to the next question. Which of the following events occur only in meiosis and not in mitosis? Separation of sister chromatids, DNA replication before division, pairing of homologous chromosome, chromosome condensation. Which may be the answer? Okay, option number 3 I am getting. Any other option? Okay, option number 3. Okay, so the answer to this question is pairing of homologous chromosome. That is option number 3. You are basically right. So coming on to the first question, separation of sister chromatids. That happens in mitosis and meiosis. Both happens in both cases, right? Meiosis as well as mitosis. And separation of sister chromatids in case of mitosis happens in which stage? At which stage of mitosis does separation of sister chromatids take place? Okay, it takes place in the anaphase, right? In the metaphase plane, the chromosomes align together at a metaphase plate at the equator. Then afterwards, the spindle and the spindle fibers are attached to a kinetochore, right? Then the kinetochore actually pulls the sister chromatid from the chromosomes to opposite poles, right? So that's where the separation of sister chromatid takes place. That is in the anaphase stage. And the DNA replication before division, that takes place in the S phase, right? Even before meiosis as well as mitosis, that happens. So that only after that it enters into meiosis or mitosis respectively. Pairing of homologous chromosome, and that is actually taking place in the prophase stage, right? Prophase 1 of meiosis, and it is not happening in case of mitosis. It consists of these leptotin, zygotin, diplotin, and dikinase, right? So this pairing, this crossing pairing starts to take place in zygotin stage, and the crossing over happens in case of by the formation of synaptominimal complex, and it leads to formation of bivalent, what is being seen in case of packeting stage, and then afterwards crossing over takes place in this packeting, and then that's how the pairing of homologous chromosomes take place, right, by recombination and all, leading to what is called as a process called as crossing over. Chromosome condensation happens in prophase stage of both meiosis as well as mitosis, so that's not an option also. So in this case, pairing of homologous chromosome is seen only in case of meiosis and not in mitosis. So I hope this is clear. So anyone has any doubt regarding this? Okay, so I think this is clear. So I will just move on to the next question then. Coming on to the next question. Which type of chromatin is more transcriptionally active? Euchromatin, heterochromatin, centromeric chromatin, or telomeric chromatin? Which might be that, sir? Which type of chromatin is more transcriptionally active? Euchromatin, I am getting the answer. Any other option? Anyone else? Okay, so you are basically right there. So the answer to this question is euchromatin. So basically what happens in that, in case of like when the chromosome is condensed, it is actually bound to this particular the DNA is bound to this histone complex there, right? So what happens is that when it is tightly bound to this histone chromosomes and all, it is what is called as a heterochromatin. So it is in a close chromatin compression. So the presence of this deacetylated histone tail methylated CpG islands and all, whatever it is, like whenever this is happening, when it is in a close conformation, what happens is that no proteins or any transcription factor can bind to the DNA. So only in this case, as this doesn't happen, because transcription factors are needed to initiate transcription, right? So if it is cannot bind to this DNA because of this close conformation or tightly packed arrangement, what happens is that the DNA regulation or DNA transcription is getting repressed there, or it is reduced in this case. So at this stage it is called as a heterochromatin. So once transcription needs to happen, so what happens is that the methylated demethylation happens, and also some of the histone tails getting acetylated there. So in that case, the DNA, when this kind of modifications happens, the DNA becomes loosely bound to the histone complexes there. So what happens, this particular stage is called as euchromatin stage, and it is transcriptionally more active, because the chromatin that is present here is more in a kind of an open conformation, because this acetylation as well as demethylation of histone chords and other proteins that are bound to the DNA, it makes the DNA more loose and more interacting towards the transcription factors and all. So when it is more interacting towards the transcription factors, then chances are that it will be transcriptionally active. The transcription rate of the DNA at that particular region will be more active in that case. So that particular region of DNA is called as euchromatin. So that's the basic difference there, and it is more transcriptionally active than getting repressed there. So in case of heterochromatin, the DNA is in a close chromatin conformation because the histone is deacetylated there and the CpG islands are methylated there, and along with other factors that could be leading to a close conformation structure formation of DNA there, and hence it is the DNA is repressed from getting transcribed. When transcription needs to happen, the histone tail is getting acetylated, and demethylation of CpG islands of this particular proteins take place, or the DNA take place, leading to more loose conformation or open conformation, where transcription factors can bind to, and then it can initiate the process of transcription, and that particular stage is called as a euchromatin stage of DNA, and it is more transcriptionally active as compared to a heterochromatin. The two other are like the chromatin formation that is take place in centromere and telomere. So yeah, there's no particular definition to that. So answer to this question is euchromatin is the type of chromatin which is more transcriptionally active. So anyone has any doubt regarding this? Okay, so I hope this answer is clear. If you want me to repeat, you can just type in the chat box, or you can unmute yourself, in that case. So I will just move on to the next question then. Coming on to the next question. Which stage of meiosis is most similar to mitosis? Prophase 1, metaphase 2, anaphase 2, or telophase 1? Which might be the answer? Which stage of meiosis is most similar to mitosis? Okay, I am getting option number 3. Any other option? Okay, so I am going with the option number 2, but I am also thinking option number 3, but I am also thinking option number 2 is also the right answer, right? So if you compare both meiosis 1 and meiosis 2, you can see that in the meiosis 1, the prophase 1, if you go into option number 1, the prophase 1, you can see that the other stages like leptotin, zygotin, diplotin, diakinesis, etc. take place, right? So in that case, it is not more probably similar to that of what you see in the case of metaphase. I will just give you another diagram here. Yeah, so I think this might be more probable. Okay. So you can see that in the prophase 1, it is completely different from that of like what you see in case of mitosis, because stages like leptotin, zygotin, diakinesis does not take place in mitosis. It only takes place in meiosis, and that's why in the previous question we have discovered that crossing over or homologous recombination is only found in meiosis and not in case of mitosis, right? And if you go into the next option, what is that? If you go into telophase 1, you can see that in case of telophase, in case of mitosis, you can see that the individual chromosomes are present like this in this case, right? So in case of telophase, the chromosomes are condensed and it is not getting separated. The individual sister chromatids are not getting separated in case of telophase 1. But in case of telophase in mitosis, the individual sister chromatids are getting separated. So that's a difference that is taking place in case of like both, what you say, prophase 1 as well as telophase 1 of meiosis 1. So if you look into meiosis 1 stage of each germ cell, it is not similar to that of what is seen in case of mitosis. It is totally different there because the chromosomes are not getting separated there. So anaphase of mitosis will not be similar to that of anaphase 1 of meiosis 1. But in case of metaphase, you can see that the chromosomes are there. The spindle fibers are attached to the centromere, and it's aligns at the metaphase stage. So that stage is also similar to what you see in case of mitosis. And also in case of anaphase 2, you can see that the sister chromatids are getting pulled to the poles, right, by spindle fibers and all. It is attached to a kinetochore, you can see that, and you can see that it is getting pulled to the poles, right. So in both the cases, you can see more or less it is similar to each other. However, if you differentiate between each other, anaphase 2 will be most probably the answer from the what you say from the definition, but metaphase 2 could also be an answer to this particular question, because if you look into the meiosis 2, all the stages are more or less similar to that of except that of telophase 2, if you, because there are 4 protocells that are forming there, right. But in that case also you can see that it is more or less similar to that of mitosis. Both the meiosis 2 stages are similar to that of mitosis. So I'm going ahead with anaphase 2 and metaphase 2, but do check it out also whether it is a where there are slight differences among the two, that might be differentiating them or comparing them more similar to each other to mitosis. So do check that also. So anyone has any doubt in this case? So I had a little doubt in this case, that's why I gave both the answer. So anyone has any response to this, or should I repeat it once more? Okay, so I hope this answer is clear. Do check the answer also in this case. It could be both of them or just anaphase 2. Okay, so coming on to the next question. Yeah, nuclear membrane disintegration starts at which stage of prophase 1? Leptotin, diakinesis, packaging, or zygotin? Which might be the answer? Nuclear membrane disintegration starts at which phase of the prophase 1? Okay, I am getting option number 4. Any other option? Okay, so I am going up with option number 2, diakinesis, because this is the stage, right? Because leptotin, zygotin, packetin, and diplotin. So there is no formation that this happens within the nucleus there. So in case of this, you can see that the chromosomes are replicated and condensed, and also in case of zygotic stage, synaptic complex starts to form, and this process completed within the packetin, where synapsis is process is over there, and what you see you can see the bivalent formation there, and crossing over take place in this stage. And afterwards, diplotic stage, the crossing over is completed and the synaptic complex have been dissoluted. So afterwards, the nuclear membrane starts to fragment out in this case. So what happens is that afterwards, the other stage like metaphase, anaphase and telophase happens there. So in that case, the nuclear membrane needs to be disassembled or degraded out there. Only then the chromosomes there, right? The individual chromosomes can be moved into separate poles, and then two daughter cells are formed, each with having intact chromosomes there, right? So at this stage, the diakinesis stage, the nuclear membrane starts to fragment out, so that it can proceed into other phase like metaphase, anaphase, etc. And then move on to the meiosis 2 stage. So nuclear membrane disintegration starts at the diakinesis stage of the prophase 1. So anyone has any doubt regarding this? Okay, so I think it's clear. So I will just move on to the next question then. Coming on to the next question. Non-disjunction during meiosis can result in which of the following genetic disorders? Cystic fibrosis, sickle cell anemia, hemophilia, and Down syndrome. Which may be the answer? Non-disjunction during meiosis. Okay, I am getting option number 4, Down syndrome. Any other option or any other answer? Okay, so the answer to this question is you are right, Down syndrome. So basically this is a distinction there, right? So in case of meiosis 1, you can see that the chromosomes getting separated to each other daughter cells there, right? So in this case, when the chromosomes are not getting separated, you can see that the one of the cells that is formed have a 3 copies of that particular no disjuncted homologous chromosome there, and the other cell having only one. So in that case, when this pass on to other set, what you see that other copies and all, when fertilization take place and all, you can see that the end product will be having a another copy of what you called as a chromosome there. So if you are having a, if you are a diploid organism, the after fertilization and all, haploid sperm cell actually interacts with the haploid egg cell there, forming a diploid organism there, right? So if the sperm cells is actually like what you say, diploid in nature, because diploid, or it has a extra copy of one of the chromosomes there. So if it is combines, it has 2 copies there, then it combines with the 1 copy of the X cell, then it fuses together to form what is called as a 3, what you observe is a 3 copy of that particular chromosome there, right? So in that case, this Down syndrome happens in this case because trisomy of chromosome number 21, consider this as chromosome number 21 there. So instead of proper separation of homologous chromosome taking place in both cases, either in meiosis 1 or meiosis 2, you can see that there is a trisomic chromosomes there. In this case, you can see that the copy of one chromosome is actually getting added up, one extra copy is present there. That's why it is called as a, in this case, in case of Down syndrome, it is chromosome number 21. So a trisomy of chromosome number 21 is taking place because of the non-distinction of this particular chromosomes there, homologous chromosome there. In some cases, one of the chromosome, in this case, you can see that as the other cell is getting 3 copies, right, or others, the remaining cell is only getting one copy. So this particular stage is called as a monosomy there. In this case, you can see that the haploid chromosomes is maintained there, so that is basically right. But in the other cases, you can see that one cell is trisomic in nature, other cell is monosomic in nature. So it can either results in these both the cases. So in other cases, I think if monosomy of one chromosome is an example of Turner syndrome, right? As far as I remember, yeah. So different diseases are arising because of this particular non-distinction of chromosome there. So if it is trisomy of chromosome number 21, it is called as a Down syndrome. Others are basically because of other factors like autosomal dominant disorders and autosomal recessive disorders, right? Hemophilia is a sex-linked recessive disorder, right? The two are autosomal recessive disorders. So that's basically other type of disorder, not because of the non-distinction of chromosomes there. So because of the non-distinction of chromosome, Down syndrome is a particular, because take place in meiosis, Down syndrome is the disease that is caused there. So anyone has any doubt regarding this? Okay, so I think it is clear, right? So I will just move on to the next question then. Coming on to this question, which structure hold the homologous chromosome together during crossing over? Centromere, kinetochore, synaptonemal complex, and spindle fiber. Which may be the answer? Okay, option number 3 I am getting. Any other option? Okay, so the answer to this question is synaptonemal complex. You can see it's right. So basically, you can see that this is one chromosome and this is the chromosome, and it is during the zygotic stage. You can see the synaptonemal complex start to form, and it is completed within the next stage, right? Packaging stage. You can see that the certain proteins are present here, and these proteins, several proteins are present here, and these proteins together helps the homologous chromosome to interact with each other directly by formation of what you called as a synaptonemal complex there, and this particular process is called as a synapse formation there. And yeah, so and afterwards, once synaptonemal complex this complex has formed, which held together the two sister chromatids together, homologous recombination between these two arms take place, so that one of the arms is getting exchanged by the other chromosome and vice versa, leading to a process what is called as a crossing over, which we have discussed, which leads to genetic diversity and all. And afterwards, like, it can move on to meiosis 2 stage once this crossing over has happened. So that's the answer. The synaptonemal complex is the structure that holds together the homologous chromosome together with chromosome during crossing over. Centromere is present here, kinetochore is attached to centromere, and basically that is what helps the spindle fiber to separate the two homologous chromosomes from each other. So in this case, that's not the answer. So I hope this answer is clear. So anyone has any doubt? Okay, so I think this is clear, right? So I will just move on to the next question then. Coming on to the next question. What is the primary function of the nucleolus? DNA replication, ribosomal RNA synthesis and ribosomal assembly, protein degradation, or lipid metabolism? Which may be the answer? Okay, I am getting option number 2. Any other option? Okay, so the answer to this question is you are right. Option number 2, ribosomal RNA or RNA synthesis and ribosomal assembly. So basically you can see that inside the nucleus, nucleolus exists, and it is the largest structure that is found within the nucleus. Other compartments are also there, but in case of nucleolus is the largest component that is present there, and it forms around the specific chromosomal region known as nuclear organizing region. So nucleoli are composed of DNA, RNA, and proteins, and the primary function is, as we have discussed before, it is synthesis ribosomal RNA and additionally assembly of ribosomal subunit. And basically, if ribosomal RNA are present there, you can basically know that it helps in which kind of process. Ribosomal RNA and ribosome assembly are involved in initiating what kind of process? It helps to promote translation, right? Translation actually happens within the ER membrane and all, because but the ribosomes that are actually assembled within the nucleolus, right? Ribosome, it helps in the formation of this rRNA synthesis, ribosomal RNA synthesis, and also ribosomal assembly, and that take place in ER. But the actual catalyst that drives this process is the nucleolus that is found within the nucleus. So, and not only that, it also helps in plays an important role in regulating the stress response and also in cell cycle regulation and all, by synthesizing several proteins that are involved in regulating this particular process. So the answer to this question is the primary function of nucleolus is the synthesis of ribosomal RNA and also helps in ribosomal assembly within the endoplasmic reticulum, because there are two types of endoplasmic reticulum, right? Smooth endoplasmic reticulum and rough endoplasmic reticulum. So in case of rough endoplasmic reticulum, it is so called because the ribosomes are actually present within the in this particular type of rough endoplasmic reticulum there, right, and not in the smooth one. And smooth endoplasmic reticulum is actually involved in lipid synthesis, that is lipid metabolism and all. Protein degradation is done by several proteins that are present within the cell, or in the case of the proteosomes and all. DNA replication actually doesn't happen here. That yeah, in the nucleus it takes place, but it's not the functioning of the nucleolus. So I hope this answer is clear. So anyone has any doubt? Okay, so I think this is clear, so I will just move on to the next question. Coming on to the next question. Which of the following protein contributes to the second central axis of synaptonemal complex of budding yeast? So we have discussed that there is a synaptonemal complex formation there. Right? So it is formed out of different proteins that arranged in a specific manner lead to formation of synaptonemal complex there. So which of these protein contribute to the central axis of synaptonemal complex? HOP1, SIP1, RED1 or ECM11? Which may be the answer? Which of this protein contribute to the central axis of synaptonemal complex in case of budding yeast or Saccharomyces cerevisiae? HOP1, SIP1, RED1 or ECM11? Okay, so I think I will just give you the answer. So the answer to this question is ECM11. So this is a diagram that I have shown you earlier. So you can see the synaptonemal complex that is happening between. If you look into from this particular view, right, you can see that this is the structure there. So the one chromosome, consider this as the blue one there, chromosomal axis there, and consider this as the other one there. You can see that there are two chromatin loops there, from one sister chromatin axis and from the other sister chromatin axis, and there are several proteins that are present between there. So this whole contributes to what is called as a synaptonemal complex there. So several proteins are there. One is the HOP1, the light blue one that is there, and the dark blue one is represented by Z1. and CYP1 is also there, that acts as a particular linker protein between these two protein complex, that is HOP1 RED1 complex interacts with this ECM11 GMC2 complex with this CYP1 complex, that enables this interaction with each other, and it also provides some kind of flexibility to the whole synaptonemal complex there. And in the central complex you can see that there is ECM11 and GMC2. So from this option you can see that the central axis, this is the central axis there, or the central element there, that is composed of this particular protein complex from the option ECM11. And also from this picture you can see that GMC2 also enables the structural aspect of this particular central element or central axis there. And these are in turn joined to this particular HOP1 RED1 complex by a protein called as a CYP1, which is flexible in nature. And this particular complex there, right, you can see this particular complex actually interacts with this particular chromosome axis there to form what is called as a synaptonemal complex. So this is the overall interaction that is taking place within, or the proteins that make up a synaptonemal complex. Yeah, so and afterwards once synaptonemal complex has formed, crossing over takes place there, right? So that's the basic theory basically, or experimental evidence there. So in this case ECM11 is the protein, one of the protein that contributes to a central axis or central element formation of synaptonemal complex in case of Saccharomyces cerevisiae or budding yeast. So I hope this answer is clear. So anyone has any doubt? Okay, so I think this is quite clear. So I will just move on to the next question. Coming on to the next question. Which protein network provides structural support to the inner nuclear membrane? Actin filaments, microtubules, lamins, or centrioles? Which may be the answer? Yeah, I am getting option number 3. Any other option? Okay, so basically you are right. The answer to this question is lamin. So basically it actually exists as a if you look into a individual structural unit of lamin, it exists as a monomer consists of this particular structure. You can see that I'm not sure whether it's clear to you, but it consists of a N-terminal head domain, consists of a flexible tail there, and a central rod domain and a tail domain consists of IG domain there. So based on this monomer actually interacts with each other to form what is called as a dimer. You can see that it is intercoiling with each other to form a flexible dimer there, or coil dimer. You can see that this is one unit of this monomer, and this one unit of monomer interacts together to form a dimer there. And this dimer interacts with another dimer at the head in a head to tail assembly to form what is called as a protofilament there. So you can see that this is a protofilament. Two dimers actually interact with each other in a head to tail manner to form what is called as a protofilament there. And this protofilament, several of these protofilament come together and intercoils or interact with each other in a parallel sheet-like manner to form what is called as a polymer there. So this is a protofilament there, which is made up of a dimer of a dimer there, and these several protofilaments comes together and interacts with each other to form in a parallel shape structure called as a polymer there. And this polymer interacts within an intercoiled manner to form what is called as a filament there. So this is a lamin filament there, and this lamin filament is what forms a nuclear membrane there. So it actually looks like a meshwork there, right? So that's why from the structure, because of this high coiling of this lamin, that's why it looks like a meshwork. Lamin forms a meshwork called as a nuclear lamina, which lines the inner aspect of the inner nuclear membrane and provides mechanical stability to the nucleus. So in between them, because of the formation of this meshwork-like structure, it forms as a barrier for the entry of substance in and out of the nucleus, and only in the presence of nuclear pore complex, which are present within this lamina, the substance can move in and out of there. So this is basic structure of lamina. It starts with the monomer there, and two monomer combines together, coalesce together to form a dimer, and two dimer interacts in a head to tail manner to form what is called as a protofilament, and each protofilament then interacts with each other in a parallel sheet like manner to form what is called as a polymer, and the polymer actually intercoils with each other and forms a tight meshwork like structure called as a filament there, which is obviously represented as a nuclear lamina there. So this is the overall structure of this nuclear lamina there. So actin filaments are not part of this inner nuclear membrane. Actin filaments are something that is attached to a nuclear lamina, but it doesn't form a part of the or a structural component of the nuclear membrane there. It just keeps the nucleus in place. Actin filaments keeps the nucleus in place, and also it helps as the helping as a network for the transport of cargo from to and fro from one place of the cell to the other. Microtubules is not present there. It is also attached to it also attaches to lamina, but however in case of mitosis and meiosis it is present there, not in case of maintaining the structural integrity of nuclear membrane and all. Centrioles are actually a component of centrosome which helps in the formation of spindle fibers or microtubules there, which enables metaphase, anaphase transition, etc. Movement of chromosomes, sister chromatids to the poles, etc. So lamins are the ones that provides the structural support to inner nuclear membrane. So I hope this answer is clear. Anyone has any doubt? Okay, so I think it's clear, right? So I will just move on to the next question then. Coming on to the next question. Which structure regulates the exchange of molecules between the nucleus and cytoplasm? Nucleolus, nuclear pore complex, endoplasmic reticulum, Golgi apparatus. Which might be the answer? Yeah, I'm getting option number 2, nuclear pore complex, or NPC. Any other option? Yeah, basically other options are incorrect, right? From the others are organelles basically. So the answer to this question, you are right, nuclear pore complex or NPC. So basically this is a structure of the diagram of a nuclear pore complex. You can see that it has a complex structure there, right? So it starts with the cytofilaments that extend to the outer end of the cytoplasm there, and it has the tendency to interact with the cargo complex or other proteins that are present in the cytoplasm, and other regions are present here. You can see that there is a membrane layer that interacts with the nuclear lamina there, or nuclear membrane there, and also there is a scaffold layer that is present in between, and also there is a Fg nucleoporine layer that is present at the central cavity of what is called a central cavity or opening of the nucleoporins. So the other place of the membrane is actually closed, but this nuclear pore actually helps as a channel there. You can see that there is a opening there, right? Several proteins are protruding out of the Fg nuclear porine there, and NUP means nucleoporine. So you can see that there are several proteins that are protruding out that interact with several cargo proteins there, and if you look into inner nuclear aspect of nuclear portion of the nucleoporins, you can see that there is a nuclear ring there, followed by nuclear basket there. So these are proteins that are arranged in a specific structure, which helps in the transport of these proteins, and this nuclear basket is actually connected to this nuclear ring by a nuclear filament there. So this is the entire structure of a nuclear porine. So it is a large protein structure embedded in the nuclear envelope of eukaryotic cell. Yes, because in eukaryotic cell nucleus is present there, right? And it is membrane bound, serving as the primary gateway for transport of molecular transport between nucleus and cytoplasm. So it's not like a passive movement of some substance can take place in nucleus, but majority of movement take place through nuclear pore complex. That is a different thing, and it consists of approximately 1,000 individual protein molecules, primarily called as nucleoporins. One of the example is nuclear. This is scaffold protein is also a nuclear protein, and Fg nuclear protein is also there, and one other is also there, membrane nuclear protein is also there, which are organized into distinct structural components. That's why you can see that the particular arrangement of each layer is seen there, or nuclear protein is seen there. Nuclear and NPCs consists mainly of 35 different types of nucleoporins. These include scaffold nucleoporins with the alpha cell node and beta propeller fold. Scaffold, you can see in this case it maintains the structural aspect of the NPC, and also transmembrane nucleoporins that anchor the NPC to nuclear envelope. This membrane layer is also called as a transmembrane nucleoporins that anchors the this NPC to the membrane, and also FG nuts that are present inside, which is characterized by phenylalanine glycine repeats. This protein, you can see that this is S-proto-Rho, and this repeats mainly consisted of this particular phenylalanine glycine repeats, and it has a cylindrical structure. From the structure you can see that with an outer ring formed of 8 subunits encircling a central channel. This cytoplasmic filament consists of 8 subunits there, and it also features cytoplasmic filaments and a nuclear basket that is extended into a nucleus. In the cytoplasmic end, it consists of filaments, and in this nuclear end, it consists of nuclear basket along with this nuclear filaments. So that's the basic difference there if you look into a structural aspect of a nuclear pore complex there. So it regulates the exchange of molecules between nucleus and cytoplasm, nuclear pore complex or NPC. So I hope this is clear, or should I repeat it once more? Okay, so I think it's clear. I will just move on to the next question then. Coming on to the next question. Recombination independent pairing of chromosomes are seen in which organism? Humans, Drosophila, C. elegans, both 2 and 3. Which might be the answer. In each and everything, when we are learning the basic, we have actually gone with the homologous recombination pairing of chromosomes, right? It is dependent on homologous recombination. In this case, it is independent of recombination. So in which organism does it take place? Humans, Drosophila, Caenorhabditis elegans, or both 2 and 3? Which may be the answer. Homologous recombination independent pairing, chromosomal pairing, which organism? Okay, so answer to this question is both 2 and 3. So in case of Drosophila and Caenorhabditis elegans, this recombination is not that important for chromosomal pairing and all. In case of humans it is important, homologous recombination is to take place, but in case of Drosophila and Caenorhabditis elegans, sometimes recombination independent chromosomal pairing are also seen. So answer to this question is both 2 and 3. So there is some difference in case of yeast as well as Aspergillus nudulans and all. So some of the species actually show this recombination independent pairing also. Yeah, so that's the answer there. So both Drosophila melanogaster as well as Caenorhabditis elegans, not in case of humans. Okay, so anyone has any doubt there? Okay then, so I hope it's clear. So coming on to the next question. Pick the incorrect statement about bivalence. They contain 2 homologous chromosomes with 2 sister chromatids. They are linked together by chiasmata. They appear during zygoting. None of the above. Bivalence formation. Okay, I'm getting option number 4. Any other option? Okay, so answer to this question is you are right. Option number 4, none of the above. Great. So you can see that in case of this is the diagram, I'm just repeating. Bivalent formation actually take place starts within the zygote stage, but actually is complete within the packeting stage. So it's not like the packeting stage they appear during they appear. But in case of zygote, they starts to appear because of the formation of this synaptonemal complex there. But it is completed within the packeting stage where crossing out take place there, right? So this option is right. Option number 3 is right. They appear during zygote, and they are linked together by chiasmata. You can see that this is the synaptonemal complex is also called as chiasmata, right? And they are linked together by this structure. So that option is also called as this option is also correct there. And they contain 2 homologous chromosomes. Yeah, it consists of 2 homologous chromosomes that are joined to form by a structure called as chiasmata or a synaptonemal complex there, right? So they consist of sister chromatids because they are homologous chromosomes there. So if you look into these 3 options, these 3 options are correct if you look into a diagram and all. So if you choose one option, that will be none of the above, because all the options are correct and none of the above is the incorrect option there. So I hope this answer is clear. So anyone has any doubt? Okay, so I think this is clear. So I will just move on to the next question then. Coming on to the next question. Which of the following disease is not a consequence of improper chromosome segregation? Alzheimer's disease, Down syndrome, Turner syndrome, or none of the above? So the following disease is not a consequence of improper chromosome segregation. I am getting option number 4. Any other option?
No.
Okay, so this simple question, right? So answer to this question is option number 1, the Alzheimer's disease. The other 2 are Down syndrome. We have discussed that before. It is because of the trisomy of chromosome number 1. So non-distinction of chromosome takes place, leading to extra copy of chromosome, the chromosome number 21 that is present within the haploid cell there, right? So in this case, like because it happens because in case of it is not sex dependent, it is sex independent in nature, and it alters the normal development of the body and brain, and it can lead to a range of physical and intellectual challenges there, because there is additional copy of chromosome number 21 there. All the other chromosomes are maintained in a disomy stage, in a diploid stage, but in case of chromosome number 21, another extra copy is there, and that happens due to either disjunction, that is non-disjunction of chromosome that is taking place either in meiosis 1 or meiosis 2, that we have discussed earlier in the previous question. And the Turner syndrome is another syndrome that is because in this case the 45th chromosome there, right? 45th chromosomes is actually designated as 45. It should be like 46, like there are 46 chromosomes that are present in case of humans there, right? It is diploid in nature, so there are 23 chromosomes there, and 22 plus 1 is the sex chromosomes there, right? So if one of the it actually happens within the female, because the female should have a 45 what is somatic chromosomes plus 2 copies of what you say plus a copy of or 2 copies of the sex chromosome there, that is XX there, right? So if one of the X chromosome is missing there, then it leads to a formation of what is called as a Turner syndrome. It is also due to like this non-distinction or any other abnormality that is taking place within meiosis 1, where the one extra copy, that is meiosis 1, where one extra copy is getting transferred into the whole cell there. So one of the cell doesn't have that particular set of chromosomes there. So that leads to absence of that particular chromosome there, which leads into meiosis 2. Other chromosomes get disjuncted properly, but this S chromosome is absent in that cell, so it cannot be separated into two other haploid cells. So that leads to a formation of this particular what that leads to Turner syndrome with this particular chromosome, that is 45 somatic chromosomes plus only one copy of S chromosome that is present there. So that is detected as either as 45 plus X or 45 plus XO or X0 there. So that's what Down syndrome is. One is the trisomy in case of Down syndrome. Another one is the missing copy of one S chromosome there, S chromosome there, leading to formation of Down syndrome there. So I hope this answer is clear. So anyone has any doubt? Okay, so I think it's quite clear, right? You also know the basics. So I will just move on to the next question. Okay, which of the following statements are correct? In mouse spermatocyte, centromere coupling is followed by synapsis. In yeast, centromere coupling is followed by synapsis. Or centromere, option number 3, centromere pairing is not important for recombination in both the species, or both 2 and 3. Which might be the option? Which of the options are correct? Read the question clearly and then answer. Okay, I am getting option number 4, both 2 and 3. Any other option? Yeah, so basically you are right. So excuse me, like I think, yeah, option number 2 is correct answer. In case of yeast, centromere coupling is followed by synapsis. So you can see that in the option number 1, in mouse spermatocyte, cell coupling is followed by synapsis. So this is one of the snapshot that I taken from the video lectures that are there. So in case of mouse spermatocyte, you can see that centromere coupling is not taking place there. So in this case, you can see that the two centromeres are separated from the homologous chromosomes are separated from each other. So it is not taking place in case of centromere coupling is not taking place in case of mouse spermatocyte. But afterwards, synapsis take place without the centromere coupling, followed by centromere pairing and other stages that are taking place in meiosis. In case of yeast, if you look at the second option, you can see that centromere coupling do take place in this case. You can see that it starts with the centromere coupling, then the synapsis forms, followed by centromere pairing, and then entry into the metaphase stage. Prophase are completed, prophase 1 has completed, then it enters into the metaphase stage. So the option 2 is correct, option 1 is wrong basically in this case. Coming on to option number 3, because we have to prove option number 4 is also correct, right? Whether it is correct or not. Centromere pairing is not important for recombination in both the species. So in case of yeast and in case of mouse spermatocyte, you can see that from the image you can see that centromere coupling is not present there. However, in case of yeast, centromere coupling is what initiates the synapsis formation there. So in case of mouse spermatocyte, no centromere coupling is there. Synapsis forms directly, followed by synapsis centromere pairing. But in case this centromere coupling take place first, followed by the synapsis formation, and then the centromere pairing. So if you look into option number 3, that is also wrong. So if you look from that point of view, if you look into this, option number 2 is correct, option number 1 and 3 are wrong. So option number 4 is also wrong because 3 is wrong, however option number 2 is right. So coming on to its answer, in yeast, centromere coupling is followed by synapsis. You can see from the diagram there. So yeah, so in that case centromere pairing is important, but centromere coupling in both the cases are basically not not important. In case of most spermatocyte it is not needed, but in case of yeast it is needed. However, synapse formation and centromere pairing are important so that it can progress into it can complete the prophase stage. Crossing over can happen. Prophase stage 1 is completed, then it can enter into metaphase 1, and afterwards completion into metaphase 2 and form the haploid cells and all. So I hope this answer is clear. So anyone has any doubt? Okay, so I think I will just move on to the next question. Coming on to the next question. In Saccharomyces cerevisiae, IME2 promotes entry into which phase? Mitotic S phase, meiotic S phase, meiotic G1 phase, or mitotic G1 phase? Which may be the answer? IME2 promotes entry into which phase in Saccharomyces cerevisiae? So I think I will give you the answer. So option is meiotic S phase, that is enter into meiotic replicative phase of DNA there. So basically IME2 is required for the timely active initiation of pre-meiotic DNA replication, that is the S phase, right? So it acts as a before meiosis 1 has started, DNA should replicate, only then homologous chromosomes are particularly formed there, right? Meiosis 1 happens, then meiosis 2 happens. So IME2 is required for the timely initiation of the pre-meiotic DNA replication. It acts as an upstream of several key regulatory pathways, similar to how this cyclin 1,3 or CDK functions in case of mitosis, because these cyclins are required for entry into like what is a S phase of this particular mitosis, right? Similarly, IME2 is also similar to this particular function also. It influences the degradation of SIC1, which is a CDK inhibitor. It inhibits several kinases there, which is critical for the transition into S phase. So SIC actually inhibits the transition into S phase. IME2 what it is does is that it acts as a kinase there. It actually helps the even though it is having a kinase-like activity, it indirectly inhibits the SIC1, and then when this particular inhibitor is inactivated there, the CDKs become active, and that it promotes the transition into S phase, and after S phase it can enter into a meiosis phase and all. Yeah, so as it is a kinase, even though it's a kinase, it doesn't directly phosphorylate SIC1, but indirectly inhibits other activities that relates to a degradation of SIC1, and then entry into this particular meiosis S phase there. And it also affects the expression of genes. Might be that it could also act as a transcription factor. It also regulates some other transcription factors that could affect the expression of certain genes that are critical for meiosis, including indicating its role in regulating meiotic gene expression at transcriptional level. So that's how my IME2 function. It degrades out SIC1 by regulating several transcription factors, or phosphorylating transcription factors that could produce gene that could actually degrade out this SIC1, which is CDK inhibitor, which then enables the transition from this G1 phase into meiotic G1 phase into a meiotic S phase. So yeah, it's like a checkpoint mechanism there. Cyclin, I think in cyclin D actually promotes that transition, right, in case of mitosis. Yeah, so that's the basic answer there. IME2 in case of Saccharomyces cerevisiae promotes the entry into meiotic S phase from the meiotic G1 phase. So anyone has any doubt regarding this, or should I explain it once more? Okay, so I think this answer is quite clear, right? So I will just move on to the next question then. Coming on to next question. Classical nuclear localization signal or NLS is recognized by importing, exporting, lamina or all the above. So each protein that should enter into a nucleus have a particular signal that is present there. So that is what NL stands for, that is nuclear localization signal. So which of the protein actually recognizes this signal so that it can get transported into a nucleus? Whether it is exportin, whether it is importin, or whether it is lamina, or all of the above. Yeah, I'm getting option number 1. Any other option? Okay, so answer to this question is basically you're right, importance, right? So nuclear localization signal is something that is found within protein that has to enter into the nucleus, right? So from that perspective, even if you look into that, you can see that the option 2 is incorrect because it is something that exports something out of the nucleus, right? Exporting. Lamina is just a part of nuclear envelope. It doesn't interact with any sub, right? And so all of the above is, from that perspective, if you look into it, all of the above is also incorrect. So the only option that is correct is importing. So basically importing consists of two different complexes, importin alpha and importin beta. So importin alpha is the one that recognizes this particular nuclear localization signal that is found in a protein. This is the nuclear localization signal there. You can see several registers there, and this is the protein. I think hope is clear. And importin alpha recognizes this nuclear localization signal and binds to it. And afterwards, importin beta, when this found this importin alpha and protein complex or cargo protein complex, it binds to it and acts as a signal protein that helps the recognition by nuclear pore complex. Once this particular cargo complex, that is importin alpha, importin beta, as well as the protein complex is recognized by nuclear pore complex, enters into the nucleus. So afterwards, when it enters into the nucleus, another protein called as RAN GTP disassembles this protein, leaving the protein that is found, which was earlier found in the cytoplasm, in the nucleus, free from the importin complexes. And you can see that the importin alpha is getting released there, importin beta is also getting released there, however the structure is not shown here, leaving behind the protein complex or the cargo complex inside the nucleus there, through the nuclear pore complex. And in case of exporting, you can see that is a different process there. The cargo complex that is found within the nucleus or inside the nucleus is recognized by this exportin. It also consists of this nuclear export signal sequence that is recognized by exportin, and similarly RAN GTP binds to it when this particular complex is formed there, and then along with the RAN GTP and this exportin like exportin complex, the cargo complex is released from the nuclear protein complex or NPC to the outside of the outside of the nucleus, that is the cytoplasm, where they are separated out. One thing you have to remember is that in case of importing, when the cargo is need to be imported, the RAN GTP is found interacting with only inside the nucleus, while in case of nuclear export, you can see that the RAN GTP is found in both inside the nucleus as well as the outside the nucleus. So there's a difference there. So the nuclear pore complex can enable both in and out mechanism, that is export as well as import there. So this nuclear localization signal should be there, and this nuclear localization is not for both exporting as well as importing, it is only for importing. But in case of exporting, the nuclear export signal is present there. So signal sequence changes or amino acid sequence changes are there, which are present there. So yeah, that's what determines whether the cargo should be exported out or imported into the nucleus. So answer to this question is the classic NLS or nuclear localization signal is recognized by importing. So anyone has any doubt regarding this? I hope this clear. So everyone needs to repeat this out, or should I move on to the next question? Okay, so I think it's clear. So I will just move on to the next question. Coming on to the next question. Which component of nuclear power complex or NPC is involved in regulating the selective permeability of cargo entering it, that is importing and getting imported into a nucleus? Transmembrane nuclear proteins, FG nuclear porins, scaffold nuclear porins, or all of the above? Which enables the selective permeability of cargo, that is importing the particular cargo into a nucleus in case of nuclear power complex or NPC. Which part of the power complex does that? Okay, so I think I will just give you the answer. So answer to this question is FG nuclear porins. So nuclear, as we have discussed earlier, nuclear pore complex consists of several nuclear porins, right? One is the transmembrane that interacts the nuclear envelope with the nuclear porins. Next is the scaffold layer or scaffold nuclear porins, which helps to maintain the overall structure of the nuclear porins there. And another one is the FG nuclear porins. So you can see that several protrusions are coming out from the FG nuclear porins, and this protrusions actually interact with the promote the selectivity. First it is recognized by the cytoplasmic filaments, then after when it needs to be entered into the nuclear pores, it checks whether the cargo protein is cargo complex is entirely maintained there, that is the cargo protein that consists of the nuclear localization is interacting with the important alpha or important beta. So this is actually this selective permeability is actually maintained by this FG nuclear porins there, or FG nuclear porins subunit of this nuclear pore complex there, which allows the entry importing of the complex from inside to outside. And not only that, it also allows the exporting of this particular proteins with a nuclear localization single layer or nuclear export single layer, and that is also the permeability, selective permeability, that is because of the FG nuclear pores that are present there. It should have a nuclear export signal there. It should have that RAN GTP that is bound to it, and along with the exporting complex there. So that particular complex is checked by this FG nuclear porin in both case of export as well as import. So that selective permeability is provided by this FG nuclear porin. Transmembrane duct actually interacts with the nuclear envelope with the nuclear pore complex. A scaffold nuclear porins actually provides the structural, maintains the structural integrity of the nuclear pore complex there. Nothing else. So only FG nuclear porins maintains the selective permeability. So I hope this is clear. So anyone has any doubt? Okay, so if it's clear, I will just move on to the next question. Coming on to the next question, pick the incorrect statement about RAN. It is a monomeric GTP binding and hydrolyzing protein. RAN works with importing. RAN can exist in two different conformational states. RAN is an ATPase. So it is something that we have discussed with the nuclear pore complex, right? So you might know the answer, which is the correct answer. Sorry, which is the incorrect statement out of this four. Yeah, I'm getting option number D. Basically, you are right. RAN is an ATPase. No, it is not an ATPase. It is a GTPase. You can see if you look into a structural component there, right? So it is a monomeric protein. Basically, it consists of only one subunit. It can have the ability to bind to GTP. When it binds to GTP, in order to promote the the transition from the either the release of the imported cargo from the complex, it needs to undergo some hydrolysis and all. and also when it gets exported into the exported out of the nucleus into the cytoplasm, you can see that in order to disassemble the whole complex from the cargo protein, you can see that the GTP needs to get hydrolyzed into GDP. So one inorganic phosphate is removed there, and from that energy released, it is getting free. The cargo protein is getting free. So it is a monomeric protein having the capability of binding to GTP, and when it binds to GTP, it can actually hydrolyze proteins. So that's basically a right answer there. And RAN works with importing. So if you look into a statement, it works with importing. So that's basically correct. But it can also interact with exporting there, right? So RAN can work with importing as well as exporting. But with the statement point of view, this statement is correct there. And RAN can exist in two different conformational state. Yeah, basically it can exist between two different conformational state. One is the GTP bound state, and another one is the GDP bound state. In case of so when it is getting hydrolyzed, it gets converted from a GTP to a GDP bound state. RAN is an ATPase. No, it is not an ATPase. It is a GTPase. It has the capability to hydrolyze GTP. It has the capability to bind to GTP and then hydrolyze into form a RAN GDP complex there, right? So that is the incorrect statement there. All the other statements are correct. So I hope this answer is clear. So anyone has any doubt regarding? Okay, so I hope it's clear. So I will just move on to the next question then. Okay. The Fg nuclear porins are mainly composed of which amino acid repeat? Leucine rich, phenylalanine glycine rich, tyrosine glycine rich, or phenylalanine tyrosine rich? Which might be the answer? We have discussed this before. It is required to maintain the selective permeability of nuclear complex there, right? So, however, it consists of certain protrusions, and this protrusion consists of several amino acids there. So, which majority amino acids are present in this Fg nucleoporins there, which enables selective permeability? Yeah, I'm getting option number 2. Any other option? Okay, so the answer to this question is you are right, phenylalanine glycine rich residues there. So we have discussed that before, so I am not going to repeat it. So I hope this is clear. So anyone has any doubt regarding this? Okay then. So coming on to the next question. The nuclear export signals are likely to be rich in which of the following? Leucine, serine, threonine, or histidine? Which might be the answer? So each protein has a concept of repeat signal sequence that are found at the terminal ends of that particular protein, which is recognized by, in this case, exportin as well as RAN GTP, which together forms a complex there and helps in this exporting there, right, through the nuclear pore complex from the inner nucleus to the cytoplasm. So what kind of signal sequences or amino acid might be present in this sequence? Okay, so yeah, I'm getting option number 1. Leucine, any other option? Okay, so I will just give you an answer. So answer to this question is leucine rich content there. So basically you are right. Nuclear export signals are primarily characterized by the rich content of these leucine residues, and it often consists of multiple hydrophobic amino acids, particularly leucine there, arranged in a specific pattern that facilitates recognition and binding by exportin. Not only by exportin, but also once exportin binds to it, RAN GTP binds to it, and then it is recognized by nuclear pore complex, which exports it out into the cytoplasm. And then GTP hydrolysis take place, leading to formation of RAN GTP. Once this happens, the S-protein becomes loose from the protein, thereby cargo protein, thereby releasing it out into free into the cytoplasm there. And many of the single sequences are described as being leucine rich. The studies indicating the presence of leucine is crucial for effective nuclear export. So several mutational analysis was done, which gives you an idea that this leucine rich sequence should be there, otherwise the export from the nucleus into cytoplasm won't take place. One such example is the P53 protein, which contains highly conserved leucine rich this sequence that mediates export from the nucleus, and it consists of hydrophobic factor. We have discussed that before, and other than that, leucine, isoleucine or phenylalanine is also present there, which is a part of this nuclear export signal. So I hope this is clear. So anyone has any doubt regarding this? Okay, so I think this is clear, so I will just move on to the next question. Coming on to the next question. Pairing of homologous chromosome and crossover occurs at which phase of the meiosis? Prophase 1, metaphase 1, anaphase 1 and telophase. This is a basic question. Yeah, so answer to this question is option number 1, prophase 1, right? So this I have seen, I am not repeating it. So I am going to the next question. What is a CRM1 protein that is involved in nuclear localization? Whether it is a RAN GTP, whether it is an protein alpha, whether it is a protein beta, or whether it is an S protein, which might be the answer. CRM1, which is involved in nuclear localization or nuclear transport. Okay, so the answer to this question is exporting. So you can see that there is exporting there. So one such example of this exporting protein is CRM1, which binds to the cargo and RAN GTP. So anyone has any doubt in this case? Okay, so I hope it is clear. So I will just move on to the next question then. Which one of the following is a meiosis specific kinase? IME1, IME2, RUC8 or SPOIL1? Which might be the answer? We have actually discussed this question before. So I have mentioned that it is a kinase. Okay, I am getting option number 3. Okay, so the answer to this question is IME2. So basically we have discussed this before, like IME2 is actually inhibits SIC1. It leads to degradation of SIC1. So basically it is a kinase there. SIC1 is an inhibitor of the cyclin 5 or 6 or a cyclin 1, 3 or other CDK proteins there. And it also inhibits this SIC1 inhibitor, leading to the activation of this CDK cyclin 5 6 CDK complex. And it also activates this NDT80 protein, which is actually a transcription factor. It actually acts as a transcription factor because it binds to the promoter region of both these CDK complexes or the genes that leads to transcription or translation of this particular protein complexes there, leading to its activation there. And once it is activated, DNA replication as well as synthesis or recombination take place, which is essential for meiosis. And also it also helps in activation of this CLB cyclin B1 and CL cyclin B4 complexes, which also helps in meiosis complex there. And not only it acts as an inhibitor of SIC1 by activating other transcription factors, phosphorylating other transcription factors that might be inhibiting this SIC1. It also helps in activation of this particular transcription factor by phosphorylating it in NDT80, which helps in actually activating meiosis there. So RUC8 is actually a component of coercin complexes. Coercin, you know that the chromatids are held together by these coercin complexes there. and also it maintains therefore it maintains the chromosome integrity, and it also plays an important role in homologous recombination there. And apart from that, if you look into the last option, SPO11 is initiates this recombination by promoting double strand breaks that is present in DNA there. For homologous recombination to happen, this double strand breaks should be present there, and from this one of the strand is getting linearized out. It binds to the other homologous chromosome, chromatin region of other homologous chromosome, thereby promoting recombination there. So it consists of a tyrosine residue that helps in this particular active site, particular double stranded breaks there. So I hope this answer is clear. So anyone has any doubt? Okay, so I think it's clear, right? So I will just move on to the next question. Coming on to the next question. Which of the following nuclear bodies play a role in processing and storage of snRNP or small nuclear RNPs or ribonuclear proteins? PML bodies, polycomb bodies, Kajal bodies, or none of the above? Which might be the answer? Which of these particular nuclear bodies, these are present within the nucleus other than nucleolus, helps in the processing and storage of snRNPs? Okay, so the answer to this question is Kajal bodies. So basically it is involved in processing and storaging of these snRNPs we have discussed before. So it is biogenesis and maturation of snRNPs, and are responsible which are essential component of these snRNPs are essential component of spliceosome, mRNA splicing, right? The introds need to be removed from the mRNA, so spliceosome actually comes out and carries out this process, and these snRNPs are actually part of this complex. So it is actually synthesized from this precursor snRNPs, which accumulate within the Kajal bodies, which before being transported to other nuclear domains such as nuclear specules, which are also components of this, which are also nuclear bodies, and where they participate in this splicing activities, this snRNPs. And it also helps in the quality control of snRNP assembly, and also ensuring that it is assembled properly before the splicing has started. And also it undergoes and inside this like Kajal bodies, other modification do take place, like pseudourylation, 2' or methylation, etc. In the snRNP, so that they can be functionally active and also assembly take place properly. So this is also an essential activity of this particular Kajal body. And into the other option, that is PML body or pro-myelocytic leukemia nuclear bodies, are membraneless nuclear structure found in mammalian cell. And this consists of several these PML proteins that acts as a scaffold for recruitment of numerous other proteins, and which are involved in process like as thymosylation. And it helps in the it helps to control the concentration of transcriptional activation and repressors within the nuclear chromatin environment. So it is somehow related to epigenetic as well as other modifications, right? If it is regulating the concentration of transcription activities and repressor, it is involved in that. And also it helps in regulating apoptosis by regulating the regulators of p53 genes, and also it is involved in DNA damage response and repair mechanism. So I'm just giving you some extra knowledge there. And also other option is the polycomb bodies, and these are also nuclear bodies, and they are also involved in epigenetic regulation by silencing genes as well as remodeling chromatin. This is involved in regulating the levels of activators and repressor. This is involved in epigenetic mechanisms like silencing and remodeling chromatin. And it is present within specific regions and also bind to specific genomic sequence, so that this regulation can be taking place there. It consists of several of these proteins. You not need to go to decay into PRC1 as well as PRC2. And histone acetylation or methylation take place in this case. So yeah, histone can be picunulated so that it can be reinforced the silent state and all, so that heterochromatin as well as euchromatin state can be maintained there. So that's the particular role of this particular body there, that is PML body as well as polycomb bodies. So anyone has any doubt there, or should I explain it once more? Okay, so if it's clear, so I will just move on to the next question then. Okay, During which phase of the meiosis does clustering of telomere take place or occur? Early leptotin, late leptotin, zygotin, or packeting, which might be the stage. Telomere clustering is actually responsible for recombination and everything for meiosis transition and all, meiosis 1 to 2 transition and all. At which stage does it take place? You know that it is talking about prophase 1, right? Okay, so answer to the question is zygotic stage. So at this stage, when clustering happens, the telomere clusters at the specific region near to the nuclear envelope, bind to a nuclear envelope, and this structure is called as a nuclear pocket there, where the ends of the chromosome cluster at one end of the nucleus, facilitating the pairing of homologous chromosome there, right? In zygotic stage, actually, this is essentially needed because chromosome, this what you say, chromosome crossing over need to be initiated, and that take place within the synaptomial chromosome formation within the zygotic stage, right? So this telomere actually binds to a nuclear lamina region there, main and clusters together to form what is called a telomere bouquet, and this telomere bouquet consists of these homologous chromosomes there. And this clustering begins within the late leptotin stage, overlaps with the zygotic stage and persisting by into the packeting stage, as the homologous chromosome undergoes synapsis and recombination process there. Chromosome crossing or take place in packeting stage. So it take place in zygotic, then afterwards once telomere crystalline has happened, it proceeds into this packeting stage where crossing over take place. So anyone has any doubt regarding this? Okay, so I think it's clear. So I will just move on to the next question. This is the last question. What is the role of SIC1 in meiosis? It activates cyclin B5 or cyclin B6 CDK complex formation. It inhibits that particular complex formation. It activates NPTEL or none of the above. What is the role of SIC1 in meiosis? If you know some of the previous question, you can answer this one. Yeah, I'm getting option number 1. Yeah, I will. You are basically wrong. In this case, it inhibits cyclin B5, cyclin B6 CDK complex formation. Sorry, I also told the previous one information there. So actually, IME2 is the one that activates cyclin B5 and cyclin C6 CDK complex there. It activates this NDT80, which is a transcription factor that helps in the regulation role or gene expression of this particular complex there. However, it inhibits SIC1. So if SIC1 is active, so which means that it inhibits the cyclin B5 or 6 CDK complex formation there, which can later does not enable the transition into DNA replication or synapse formation there. If DNA replication does not take place, then meiosis, it doesn't enter into meiosis there, right? So SIC1 has a function like that. So it is inhibited by IME2, which is a kinase, indirectly, and therefore meiosis transition takes place by the activation of the cyclin B5 or 6 CDK complex, while SIC1 actually inhibits it. So I hope this answer is clear. So anyone has any doubt? Okay, so I think that is cleared out. So thank you for joining for this class. This is the last question. So does anyone has any last minute question? Do you want to ask or anything about this current topic or from the previous topic that you have discussed? Okay, so I think we can end the class for today. So thank you everyone for joining. We will see you in the next class then.